Okay, so welcome to this next video in the playlist on cancer. In this video what we're going to do is we're going to talk about the PI3 kinase, AKT, and mTOR signaling pathway. So PI3 kinase, usually just denoted as PI3K, then AKT, also called protein kinase B, but we'll see all of this, and then mTOR is more downstream of that. And this is another pathway, basically, which is associated with growth factors and growth factor receptors. So in the previous set of videos, we saw the MAP kinase ERK pathway, which was one pathway which was going to be activated by growth factor binding to growth factor receptors. This is another pathway which is going to be activated by growth factor binding to growth factor receptors. Okay, so this is what we're going to do basically. If we have a cell here, and we put this cell in the presence of growth factor, so let's say this little box is going to denote some growth factor, so this is going to represent growth factor, and I might colour it, I've done an A rather than an O there, never mind. Growth factor, not growth factor. Growth factor. Okay, I don't know what's wrong with my spelling at the moment. I'm usually not bad. Growth factor. Again, we're colouring growth factor orange. Right, so we want to see how does this growth factor induce the cell to divide, and we've already seen one pathway by which growth factor can help a cell to divide, but we're now going to see another one basically, and this is the PI3 kinase AKT mTOR pathway. Okay, right, so let's take out a piece of cell membrane and show firstly the growth factor receptor. And I wanted to stress that this receptor that is going to be activated by the growth factor is the same one as we discussed in the previous videos, and in fact the receptor activates both downstream signaling pathways, and they both contribute to the cell beginning the cell division process. Okay, so let's say this is our growth factor receptor in the cell membrane, and I'm just going to draw the phospholipid bilayer here. So there's the phospholipid bilayer, and here is our growth factor receptor. So let me colour in that a certain colour. I'll colour it in orange, right, so orange. Okay, so here's our growth factor receptor, and I'm deliberately keeping it very general. I'm not giving you specific growth factors, I'm not giving you a specific growth factor receptor, because there are many different growth factors that you can name. VEGF, vascular endothelial growth factor, EGF, and epidermal growth factor, PDGF, platelet-derived growth factor. There are loads of them, and they all work in the same way, so we're going to keep it nice and general. But if you do want a concrete example, the sort of archetypal example that you could think of would be epidermal growth factor, more commonly just abbreviated to EGF. So this is epidermal growth factor. And the receptor for that would be called the epidermal growth factor receptor. And just as the epidermal growth factor is reduced to EGF, the epidermal growth factor receptor would be reduced to EGFR. Okay, so here's our general growth factor receptor, but if you want a specific example, you could think of epidermal growth factor receptor, EGFR. Okay, right, so let me bring this more into the center. Okay, so here is our growth factor receptor in the phospholipid bilayer of our cell. Now what's going to happen is when the cell is exposed to growth factor, this growth factor is going to bind to the extracellular domain of this growth factor receptor. So here is the growth factor bound to the external domain of the growth factor receptor. And basically, when the growth factor binds to the external domain of the growth factor receptor, what happens is the growth factor receptor changes in conformation slightly. So let me draw a new cartoon for this happening. So basically what changes is it changes conformation slightly so that it can now dimerize with another growth factor receptor. And don't take this picture too literally. It's I've drawn this sort of lump coming out just to illustrate that something has changed so that two of them can now dimerize together. That's the important thing, that there's a conformational change and two epidermal growth factor receptors, or just growth factor receptors to be more general, are going to now dimerize together. So let's say we've got another one over here, which has also got growth factor bound to it, so that it also has this change in conformation so that it can now dimerize. So let's colour everything in so everything makes nice and sense. So here we have our growth factor receptor bound to the growth factor, and we have two of them now. Okay, and they've both changed conformation when they bound this growth factor, and they now have available new dimerization sites, basically, which can bind with each other and dimerize. Okay, so these two sort of activated growth factor receptors are now going to dimerize together. Okay, so dimerization occurs next between two growth factors which have both bound growth factor. Okay, so let's draw this happening. Here's the phospholipid bilayer, and now they have dimerized together. So here are these two growth factor receptors dimerized together like so, which for the basis of my cartoon is just these two little sort of sticky out bits, protuberances, that have now stuck together. Okay, so here is the growth factor still bound to each of them, basically. Okay, so here's a growth factor, here's a growth factor, and here are the two growth factor receptors now dimerized together. Okay, let me just finish colouring them in, and then we'll talk more about what happens next. In fact, actually, I'll talk now whilst. Right, okay, so intracellularly, once these two growth factor receptors have dimerized together, each of these receptors intracellularly has tyrosine residues down here, okay? So let me just remind you of the structure of a tyrosine amino acid. So let's draw the general amino acid structure first. So here's the amino group up here. Then below that is the alpha carbon, and below that the carboxyl group here. Okay, so far this is just the general amino acid structure. And now the specific portion comes in when you add the R group on here. So here's a methylene here, and then you've got a benzene ring, like so. And off this benzene ring, off the third carbon of this benzene ring, you then have a hydroxyl group, and that is the structure of tyrosine. Okay, so on the intracellular domain of these growth factor receptors, there are quite a few of these tyrosine residues, basically. So there's lots of tyrosine residues here, here, and here, and here, etc. And basically what happens is a process now known as autophosphorylation, which is where the growth factor receptor that you've dimerized with phosphorylates your tyrosine residues. So this first, let's call this growth factor receptor 1 and this growth factor receptor 2, and they have dimerized together, okay? Growth factor receptor 1 has these tyrosine residues down here, and growth factor receptor 2 is going to now, once they've dimerized together, it's going to phosphorylate those tyrosine residues. It's going to add a phosphate group, which has this structure here. It's a phosphorus atom doubly bonded to oxygen with two hydroxyl groups off the phosphorus atom, and then singly bonded to an oxygen that's acquired an electron from somewhere else and has a negative charge, therefore. Basically what's going to happen is you're going to perform a condensation reaction. You're going to bring water out, and you're going to put this oxygen onto the phosphorus, and that's how you phosphorylate a tyrosine residue. Of course, it won't quite happen like this. You won't just take a phosphate group from the cytoplasm and do this. Instead, what you'll do is you'll take ATP generally, snatch a phosphate group off that, and bind it on here. Okay, and the energy that's released by the hydrolysis of ATP will drive the addition of the phosphate group onto the tyrosine amino acid. Okay, so the point is that the growth factor receptor 2 is going to phosphorylate the residues, the tyrosine residues, on growth factor receptor 1. And growth factor receptor 1 will reciprocate for growth factor receptor 2, i.e. it will phosphorylate the tyrosine residues on the intracellular domain of growth factor 2. And that process is known as autophosphorylation. Okay, autophosphorylation. And basically, once the tyrosine residues have been phosphorylated, the growth factor receptor dimer is now ready to recruit downstream signaling pathways. So let's draw the growth factor receptor with these phosphorylated tyrosine residues. Okay, so here is the growth factor receptor. They are dimerized together like so. I wish I'd left a bit more space now in between the two so that I could draw the phosphate groups a little better. Here are the growth factors, still bound on the extracellular domains. Let's colour everything in. So here are the growth factors in green. Here are the growth factor receptors in orange. Okay, right across there. And then off these tyrosine residues now, you will have phosphate groups. So you'll have these phosphorylated tyrosine residues on the intracellular domain, and those phosphorylated tyrosine residues are going to be really important in recruiting downstream signaling pathways. So let me draw these phosphorylated tyrosine residues. So I've just put little dots on them to represent phosphate groups, and the same for growth factor receptor 2 here in this dimer. Okay, there we go, and we'll colour them in pink. Okay, so now what's going to be able to happen is once you've got these phosphorylated tyrosine residues, you're going to be able to recruit downstream signaling pathways. And one of the downstream signaling pathways you can recruit is the MAP kinase ERK pathway, which we discussed in previous videos. Now what we're going to discuss is another downstream signaling pathway that these phosphorylated tyrosine residues on the intracellular aspect of the growth factor receptor dimer is capable of recruiting. And we'll do that in the next video.